faculty of the State University of New York's College of Environmental Science and Forestry. And Roxanne's going to talk to us about research in the Finger Lakes, global change drivers affecting mercury bioaccumulation in Finger Lakes food webs. I'm reading that right there. And I think that's really relevant to us because we need to be thinking about how mercury bioaccumulation and concentrations might change with climate change. So go ahead, Roxanne, take it away. And thank you so much for being willing to talk with us today.
Thanks, Wayne, and thanks everyone for attending and your interest in my talk. I just converted this to PDF, so hopefully all the slides will work here. But basically, I just wanted to say thank you. I think the consortium is a wonderful thing, and I'm so glad that we have it and to produce really important documents such as, with respect to this topic, of course, the protocol on mercury consumption advisories for sensitive populations. So thank you for your work on that. So my objective today is to convince you that the Finger Lakes are a really good tractable study system for us to understand changes that are occurring in the Great Lakes food webs that could affect mercury bioaccumulation in fish. And this is our lake trout from Skinny Atlas Lake at the I caught, and I think it has a lot to teach us about what things we might expect will change. Wayne, you mentioned climate change. I'm going to talk a little bit about that if we have time to get there, given this a little bit of a later start, but also some things like land use change, invasive species, those global change drivers that are so important for us to keep our eyes on because of their potential to change ultimately mercury accumulation in our top predators. A little bit of background about me first. I am a Great Lakes native. I grew up on the shores of Lake Ontario, and it's always been a place of great inspiration for me to look out onto the lake. My formal education in limnology began at McMaster University, and I was introduced there to research programs on wetland ecology and understanding differences in habitat degradation across the Great Lakes, but also, the beauty of all these beautiful lake systems. I moved on to Queen's University for my master's and PhD work, and during my master's degree is when I was first introduced to mercury pollution and understanding, our legacy of mercury use in our society and how legacy in mercury in sediments can remain bioavailable for decades, and understanding factors that lead to increased bioavailability to food webs. My PhD research was in Eastern China, where my fieldwork was, and what this component of my education really helped me understand was a link between mercury bioaccumulation and changes in freshwater systems, such as eutrophication, because that was such a big issue in Eastern China where I was working. We'll get to that, which has can continue to interest me in understanding those interactions. And during my postdoctoral work, when I first came to the Finger Lakes region, and to really understand, one of the questions we had was to understand lake trophic status and how that influenced mercury differences among species, which I'll be showing you a little bit about today, across all the lakes. And then, as Wayne mentioned, I started my faculty position in 2017 at SUNY ESF. I'm really happy to be here, and working on, again, limnology and bringing in ecotoxicology and environmental health. So really studying fate effects and risks of toxicants, and especially mercury, to wildlife and human health. So, we have, and I'm sure this group understands well about why we care about methyl mercury, how methyl mercury bioaccumulation and biomagnification works, and that we've identified a lot of underlying mechanisms already, the mercury science community, that influence how methyl mercury bioaccumulates and biomagnifies. I mentioned primary productivity, of course, food web structure is so important, habitat use of species, which can change, based on what's, where their food source is coming from, and of course bioenergetics, how what fish are eating affects their growth and other aspects of their biology. And so these global change drivers that we're thinking about that both directly and indirectly affect methyl mercury bioaccumulation and biomagnification are these drivers that I've mentioned: land use change, invasive species, and overall having this bigger picture of climate change occurring that can alter so many facets of our freshwater systems. And the one in particular that I'm hopefully going to have time to get to is on how climate change could be affecting lake stratification patterns and hypoxia, which obviously has an important implication for methyl mercury production. But before I get into these three kind of case studies and highlights from some of my past research and also highlights from ongoing research, I just want to tell you a little bit briefly about the Finger Lakes in case you're not from New York State and you're not as familiar with them. They are located in the Lake Ontario watershed. They're a series of 11 glacially formed lakes oriented north to south, so they flow north. They vary widely in their limnological characteristics and characteristics in their watersheds, population densities, but in general, relevant to our question here, they are hard water alkaline lakes, and some of them can be famous for being very, very deep. So on Seneca Lake and Cayuga Lake have max depths of over 400 feet. So, Cayuga Lake is 130 meters deep, Seneca Lake is 200 meters deep. But we also do have shallower lakes. So Honeoye Lake is a good example that I'm going to be bringing up later on. And that lake is only 9 meters deep, so about 30 feet. So, and the great thing about these lakes is that you can drive pretty much from Honeoye to Skinny Atlas in a day's drive. And so you can have multiple systems and do these comparative studies based on your question. They are also a really important drinking water source for over 1 million people in the region, for the cities of Rochester and Syracuse. And the Finger Lakes are relevant to our discussion today. Very important from a fishing perspective, not just economically, but also culturally. I worked with this undergrad here, Alex Gatch, my first field season on the Finger Lakes, and he really helped me understand, how important this fishing is, both as, recreational activity for him, but also as a source of food. So it's really important that we understand the dynamics of mercury bioaccumulation in these lakes. One final characteristic I just want to point out is this agricultural cover. And we tend to think about the importance of forests when we're thinking about and wetland cover, when we're thinking about mercury. But we know a lot less about agricultural effects, but we do know that agriculture is a source of nutrients to our lakes and can impact harmful algal blooms. And in that respect, there's a link to mercury through the conditions that promote methylation that can occur when we have a lot of nutrients and organic matter and low oxygen conditions. So it's something to keep an eye on, and I think it's something we don't know enough about yet. So here is a little bit of a land use map for you to get a better understanding of the lakes I'm going to be talking about, oriented from west to east, and their land use covers. So you can see Cayuga Lake is really dominated by the agricultural land cover, whereas Honeoye Lake, another lake I'll be talking about, has much more forested, very little in terms of agricultural cover. So I think that the Finger Lakes and my colleagues and I both all agree that they are a great system for studying mercury cycling in the Great Lakes watershed, especially with respect to agriculturally dominated watersheds, and aquatic invasions, which you'll see in a moment, trophic status, and also atmospheric deposition. In the Great Lakes region, of course, what happens in the regional deposition patterns for Lake Ontario and regulations with respect to that have an impact on the Finger Lakes region as well. So just a quick refresher, but I'm sure you all know this, for New York State, monitoring is conducted by the DEC, and advisories are set by the Department of Health. They are established for specific species of fish from specific water bodies, and there is a general advisory across the state not to eat more than 1 meal of fish a week from all New York State freshwaters. Specifically for the Finger Lakes region, we have that same general advisory. Up to 4 meals a month can be safely consumed by both the general population and sensitive groups. I just want to point out that Oneida Lake is not part of the Finger Lakes region, but I will also be bringing up Oneida Lake later on in the talk because it is the location of the Cornell Biological Field Station and a long-term study since the 1950s of those fish populations. So that's another system that really gives us an opportunity to understand mercury bioaccumulation and changes because we have this long-term data set on the fish food web and factors that have affected the lake over time. So the final thing I want to mention before I show you some data is this publication by Simonin et al. Howard Simonin did this statewide study to monitor mercury in New York State fish. And they did sample 4 of the Finger Lakes that we sampled. So we have some data that we can do some temporal trends analysis with. And Simonin et al., they devised some models to predict mercury concentrations in various species using pH and wetlands cover or length and pH, depending on whether you were aiming to predict mercury concentrations in a standard size fish or using all the data available. So I will come back to this as a kind of a decade later when we conducted our work to compare some temporal trends, which will help illuminate our questions with respect to those climate changes. So just to give you a sense of what kind of concentrations we're talking about here in the Finger Lakes, so total mercury concentrations in wet weight on the y-axis, and our golden shiner, bullhead, yellow perch, largemouth bass, lake trout, and walleye were some of the species we looked at. And we're going to compare them to the 300 nanogram per gram criterion. And if you see in red, the points were those fish that exceeded the guideline. And we have about a quarter of our fish sampled that exceeded that. Of course, greater exceedances for our higher trophic level fish, driving that point home that we all know so well, that food web trophic position matters. And really not as much cause for concern, of course, aside from these very high walleye here, which I'll just mention briefly, have their own story. We did determine that those to be fish all from Owasco Lake that were very old. The probability of a walleye being older than 11 years, I've worked with this on Wayne, is very, very low. So the reason why these walleye were so low is that they had been stocked in Owasco Lake, and that stocking program had been halted, and these were just some old remaining fish in the lake. So now that you have that sense of what we're talking about in terms of concentrations, I want to go into my first question, which was wanting to get to this idea of how does primary productivity impact the mercury bioaccumulation that we're seeing across these species. So the first thing we have to ask is, are there differences across the lakes? And then if so, what are those drivers? What are the factors that resulted in higher concentrations of mercury in these fish? And is trophic status one of them? So I'm going to be showing you just a quick series of graphs with size-adjusted total mercury concentrations on the y-axis, and then lakes that contain the species. Some of them, don't have the walleye or don't have the lake trout, depending on the depths. But lakes will be oriented from west to east. So what I want you to take away, these have been size adjusted to these lengths here, is that for some, lake effect varies by species. We saw no differences, for example, across the lakes in largemouth bass mercury concentrations, but we did see differences in the lake trout and the walleye. And I just want to hone in on one example of a difference that intrigued us, was this difference we saw between Seneca and Cayuga lakes, which we assumed to be, similar systems. And we'll get to what drove those differences in a moment. In forage fish, again, lake effect varies by species. We did see a difference with bullhead. Sorry, this should be a check mark here. And golden shiner and yellow perch. But in the lower food web, again, we saw that it varied depending on what you were looking at, what invertebrate group. So we didn't see differences in the zooplankton and the pelagic sources, but in the benthic invertebrates, we did see differences among lakes. And interestingly, when we looked again at that Seneca-Cayuga comparison, we found the reverse trend than what we had seen in the lake trout. The Cayuga benthic inverts actually had higher mercury concentrations, which is actually something we had expected, because I didn't mention this at the beginning, but Cayuga Lake does have a defunct coal-fired power plant on it, so we had kind of expected we would see higher concentrations in the fish as well. So it surprised us that the lake trout had this different trend compared to the benthic invertebrates. So when we put all of our results into a mixed effects model, and we ran lake as a random effect, and we put in various factors as predictors in our fixed effects, trophic status did not come out as a predictor of fish mercury concentrations, and nor did many of the other factors we put in, like lake morphometry variables or land cover variables, nor did lower trophic level methyl mercury concentrations. So what this led us to believe was that something that is driving these concentration differences in the fish has to do with food web structure in these lakes. So just to summarize there, we didn't actually find that trophic status was a predictor of fish mercury concentrations at this single point in time in the Finger Lakes region, despite quite a high range of variables. And it wasn't so surprising that we didn't find pH was a significant predictor, because the pH of these lakes is all very similar. Whereas you would find pH as a driver when you look across New York State, because you incorporate a larger range, such as those low pH lakes in the Adirondacks. So let's get back to this idea of food web structure, and what really can affect food web structure. We all know are the introduction of invasive species. And what we found in our study is that habitat use in particular can be affected by invasive species. So let's look back at those lake trout results. And one important thing to note is that Cayuga Lake since 2010 has had presence of round goby, whereas Seneca Lake still has not yet, miraculously, we have not yet detected round goby in Seneca Lake. So we hypothesized that what could be driving these lower concentrations in lake trout from Cayuga Lake is the presence of round gobies in the lake trout diets. And what this would result in would be a faster growth rate, which would allow the same amount of mercury, which is represented by these same sized mercury circles, to be diluted in a larger mass. So that was our working hypothesis. At the time, when I had presented these results to the Oneida Lake Fisheries group, the great thing about doing those talks is that one of the fisheries biologists, Randy Jackson, came up to me and said, hey, in 2010, we actually did a mercury study with Charlie Driscoll, and we have those data, and wouldn't it be great to do this pre-post invasion analysis? And I said yes, because that would help us, understand what's going on in the Cayuga-Seneca Lake example. So we took this long-term fish data set, so that includes weekly gill netting we have fish diets, we have fish age, we have fish growth for every year. And so we took the 3 years prior to the invasion, which occurred around 2013 and 2014, and we took the years 2015 to 2017, and then we did mercury sampling in the 2016-2017 to represent our post-mercury data set. So I won't have time to go into all the results, but I also just want to give you a sense of how high are these mercury concentrations relative to the Finger Lakes fish. They're pretty comparable, they're low. No fish exceeded the DOH 1 ppm guideline. We had most of the fish, only 10% exceeding 500 nanograms per gram, and about 40% of our predatory species exceeding the 300 nanogram per gram guideline. So here are the percent of fish composed in the diets of each species. So the first bar I'm showing you is pre-invasion, so 2010 to 2012. And you can see it's mostly dominated by this purple, which represents the gizzard shad. There's no green, of course, because this is pre-invasion. But when we go to the post-invasion, we see that all these species that we looked at did incorporate round gobies into their diets. They also, we also saw that they incorporated a lot more yellow perch. And in some cases, it was a large component of an increase in their diet composed of round goby. And what did this mean for their mercury concentrations? I'm showing you here mercury concentrations on the y-axis and these various species we looked at. And all the mean mercury concentrations did decline post-invasion. And it was not a significant decline for smallmouth bass, but a significant decline for walleye and white perch. And I haven't shown those results for yellow perch because a simple, ANOVA like this is not appropriate because there were significant differences in ages. So I used a mixed model approach to also look at yellow perch differences pre and post invasion, but I'm going to leave those out for time's sake. But in general, we found that the incorporation of this benthic prey item into the diets of these Oneida fish resulted in lower mercury concentrations. So we saw that this change to this benthic pathway did decrease mercury exposure, but what I haven't had time to show you is that that wasn't actually related at all to growth. We didn't find any very clear or strong signals that growth rates were different after the invasion, but rather that the collective change in the diet, which was from eating potentially, higher trophic level fish, in some cases, like with walleye, they switched to eating round goby, and which have similar mercury concentrations at the age sizes that they're eating them to other prey items. So this is a little different than what maybe you've read about in other Great Lakes systems, Lake Michigan and Lake Erie. Both predicted a higher, an increase in mercury with the presence of the round goby. So I do think there's a potential for the effect of invasive species to be really water body specific, and that we really need to really understand all the changes that are going on in the diets of these fish to really understand what the impact of an invasive species can be. How are we doing for time? 1:30. No news? Okay.
I would say 10 to 15 minutes more, if that works for you, Roxanne.
Yeah, I think I can do that. Hopefully I'm not overwhelming people too quickly with a lot of information. So in terms of climate change, one of the ways that climate change can affect lakes is by changing their stratification patterns. And so when, especially in shallow lakes, this can increase the duration of stratification if they are wind-driven. Mixing is wind-driven over the course of the summer. And the increase in hypoxia, we know, is conducive to the methylation of mercury. So one kind of insight I got from our results was this temporal comparison we did to the Simonin et al. Work. So what you're seeing here are the lake codes. The Simonin et al. Predicted mercury for a 9-inch yellow perch, and our predicted mercury concentrations for the same sized yellow perch using their model. So we saw differences, increases, decreases, no change. So really varied by lake. Again, a trend we continue to see, but no changes that were considered significant based on an error that Simonin developed of 200 nanograms per gram. So all those changes were within the 200 nanograms per gram. But when we looked at the higher predatory species like largemouth bass and walleye, what popped up to us especially was this significant increase, very large increase we were seeing in largemouth bass from Honeoye Lake and walleye. We already talked about the Owasco example being sort of artificial because it was driven by that stocking and old age of those fish. So what was going on in Honeoye Lake in particular in the last 10 years? Well, unfortunately, this. So Honeoye Lake has had a dramatic increase in harmful algal blooms and eutrophication in the last 10 years or so. Luckily, we have a long-term data set for Honeoye Lake based on a lot of work of local collaborators, but also some very active citizen scientists on the lake. And this is surprising because, like I mentioned to you, this is not a situation where we have a lot of external nutrient loading going on. The lake is largely forested, and all this, the wastewater does not go into the lake. People are on the sewage system, so it's really driven by internal loading. This is work that I'm collaborating on with Nelson Harston at Cornell University, and many other collaborators. But what we're seeing is that there's been this increase in hypoxia, really significant since 2013, which you can see in the gray bars here. And so, the hypothesis is that potentially these lakes that are deep enough to stratify but shallow enough to mix, they stratify when the lake when it's warm and it's calm weather, and this allows low oxygen conditions to become dominant, which releases nutrients into the hypolimnion. And then when the wind becomes strong enough to destratify in a storm, you get mixing of that phosphorus, which we're hypothesizing is what's potentially causing these HABs in part. It's a complicated story. But with respect to mercury, one of the ongoing work we have is to understand, is this hypoxia also resulting in more methylmercury production? And I have some zooplankton sampling that I'm working on that is to address that question. So these climate-driven increases in hypoxia can potentially increase fish mercury. And this is also a topic of research that I'm working on currently with my colleague at ESF, Karin Lindberg, who I'm a co-PI on her NSF grant, which is to use otolith chemistry and also this novel structure, eye lenses, which turn out to accumulate mercury in really high concentrations, and to try to understand if we can track both hypoxia exposure and mercury exposure in fish using these structures in Lake Erie. So these are some very preliminary data for Western Basin yellow perch. So to summarize, Finger Lakes fish mercury concentrations are relatively low, but at the same time, when you dig in a little deeper, you do see these lake-specific differences and the potential for things to go from very low to high on a short period of time, I would say in 10 years time frame. These research on these global change drivers is really necessary to predict fish mercury bioaccumulation going forward. And of course, we all know about the round goby invasion, and I'm also working on some studies looking at hemimysis. And it probably won't. These won't be the last invaders we see, and we need to be able to sort of study quickly and predict how these changes might affect mercury bioaccumulation. And I think the Finger Lakes really offer us this tractable study system to understand changes that are relevant to the Great Lakes system. Funding from this work was from NYSERDA, the Finger Lakes Institute Endowment, my startup at ESF, and the hypoxia work I just mentioned towards the end from NSF. We got a lot of help collecting those fish from the DEC, our collaborators at SUNY Brockport and the Finger Lakes Community College, and then also from some local anglers. My collaborators, Lisa Kleckner and others at Hobart and William Smith Colleges, Charlie Driscoll at Syracuse University, Randy Jackson, and Tony and Tom Lars at the Cornell Field Station, and Nelson as well, and my collaborators, Abby Webster and Deb Driscoll at ESF, for a lot of help with the mercury analyses, and then just so many students to thank for their work. And this is some of our fun lake sampling that we did for the Finger Lakes work and my graduate group, and toddler that keeps me up at night. So thank you so much for your time. I appreciate it.
Wow, thank you, Roxanne. That was really interesting. You've done a lot of work there. And so I'll certainly open that up to questions for folks.
Hi, this is Henry Splitoff from New York State Department of Health. That was a wonderful talk. It was really interesting for me, and it's particularly relevant for us because we're going to be looking at mercury data statewide in fish and regionally throughout the state, and trying to examine temporal trends and spatial trends. And so this is really helpful to see your insights into the data. I had one question. I got a little lost with when you were toward the end of your first segment, when you said that you had the hypothesis that the difference or the goby invasion, the brown goby invasion in Lake Seneca, I believe it was, or sorry, in Cayuga.
Yes.
That your hypothesis was that would result in higher feeding, higher growth rates, and dilution of the mercury, and that's why you were seeing lower mercury in Cayuga. Yeah. But then you said that you didn't find evidence of faster growth, and so.
So what was?
Yeah, I guess so.
Good question, and I'm glad you picked up on that. Yeah, I mean, I probably could have spent this whole time just talking about the Oneida study, but so what we did find, so we looked, because we have this great long-term data, and we were able to look at diets pre and post in really a lot of detail, both like how much fish they consume and which species of fish they consume, but also what proportion was consumed based on invertebrates. And I didn't show those data, but so what happened was that there wasn't a change in the amount of fish consumed, so they still ate the same amount, but they switched to round goby and yellow perch. And our data don't seem to indicate that there's a really big difference in mercury concentration among those prey items, like the gizzard shad, yellow perch, and round goby that they are eating. Once they get larger and older, yes, potentially there's significant differences there, but the ones that they're actually eating aren't that different. So what I did find was that two things are really of note, is that they did switch. In many cases, they were eating some higher trophic level fish as well in this other fish category, which would have included like walleye were eating some small, juvenile smallmouth bass or other walleye. And now they were moving entirely to eating these lower trophic level items. So that's one thing. So a shift not just to the benthic pathway, but also from higher trophic level to lower trophic level species. And then the thing that just happened at the same time was that there was an increase in mayfly consumption by fish. And they've seen this steady and continued increase in mayflies, which is actually what was unusual was the loss of the mayfly for some period of time, but the mayfliers are back. And so those, of course, would be also a lower mercury item compared to fish. So I think we have to look at the whole diet, not just at what single species and how much of it is being eaten, but how all the changes in diet have occurred.
Okay, thanks for explaining. It's
Yeah.
interesting.
Yeah.
I may reach out to you in the future for.
Sure, please do. And I'm hoping to have that paper submitted soon. So I can also share that with you once it comes out, if you send me your contact info.
Okay, sure, I'll do that. Thanks.
Thank you for the question.
Any other questions? So I will throw out a comment, and when you started talking about the same topic that Henry just raised, when we did our sort of retrospective historical look at mercury in Lake Ontario fish, over the last 20 years or so, we didn't see much change except in smallmouth bass, which went down. And there was a big diet change because of the round gobies coming in, and also the age and size went. Let's see, they got bigger faster, and their condition factor got better. So it's much more of a hand waving argument. But I mean, you really point out the complexities here and need to understand. So I really appreciate the detail here.
Yeah. Yeah, I mean, it really surprised us that we haven't yet seen those changes in growth or condition in the Oneida Lake food web. And, maybe it's because it's just too soon after the invasion. Maybe, we'll start to see that. But so far, we haven't seen that change in growth, which we really did expect. We do have another project ongoing with some collaborators, again, at the Lisa Kleckner at the Finger Lakes Institute and collaborators at Cornell University that are leading this bioenergetic study. So looking at Seneca, Cayuga, Oneida, Lake Ontario, and trying to better understand these bioenergetic components to the story as well.
Good. Wow. Others? Questions? Anyone else?
I guess I'm curious if anyone, I mean, obviously the people from Lake Erie are dealing with the hypoxia issue, but one of my colleagues, John Farrell, works up at the Thousand Islands in the St. Lawrence River, and they're starting to see some hypoxia there too in shallow areas. So I'm curious, how much maybe some people if that's on their radar in terms of something that could be increasing mercury or.
Yeah, it's not something I had thought about. This is an eye opener for me.
Yeah, and the example I give with honey white is different than in Lake Erie. It's nutrients coming in externally into the lake. And driving that hypoxia. But in these shallow lakes that have a lot of internal nutrient loading, the stratification patterns really do matter, and those could potentially be changing as the weather warms and the warmer water at the top really gets harder to mix as the temperature-driven changes are more stable.
Right.
This is John Estenek from Ohio. This is John Estenek from Ohio EPA. Susan Fisher published a paper looking at transfer of mercury goby to bass in western Lake Erie some time ago. So I would imagine if you're not familiar with that paper, I'm sure you might want to take a look at it.
Was the first author Hogan? Is this a 2007 paper?
I'm not sure of the date. It could
Okay.
be back at 2007. It
Right.
was Susan Fisher and a number of other authors.
So if we're talking about the same paper, I think that what happened in Lake Erie was that over the course of the time of the invasion, there were also decreases in the atmospheric deposition and inputs of mercury into the lake. And so they didn't actually see a change in the mercury concentrations of the smallmouth bass pre and post invasion, despite this much lower availability of mercury. And they also did see, I believe, an increase in growth of the fish. So they assumed that this change in mercury that was or the expected change would have been a lot lower, but they didn't see that, and so they assumed that that was the contribution of round goby to the diets. And so sort of the going hypothesis is that you have these mussels that are accumulating particulates and mercury from the water column, but they were previously obviously not available to the food web. But with the invasion of the round goby, that exploit those dry synid mussels, and then fish being able to consume the gobies themselves, this makes that pathway of potentially of toxicants available to higher trophic level fish. At least in Oneida Lake, the size of round goby that the fish are eating are small, and the size of the zebra mussels that the fish are eating are small. So it's potential that these are just not haven't had time to accumulate a lot of mercury from the water column. So as even though yes, they could be a potential source, and we have to take those kind of factors into consideration as well. But I will make sure, John, that I have the right paper. I'm talking about the right paper. I hope so, that it's the same one. Thanks for that.
All right.
Any
other questions? Oops, sorry. Go ahead, Roxanne.
I'll just I'll just add one other point that I think is important, and I'm sure this group has thought a lot about this, but some of my work in China looking at trophic status, one of the things I was really interested in understanding, because at least in Eastern China, fish consumption is a really big part of the diet, was, yes, the risks are from methyl mercury, but what are the benefits from fatty acids? And what are potentially any protective effects of selenium present? But I wanted to ask these questions in the context of looking at these differences in trophic status. So would trophic status and primary productivity potentially influence the availability of some of those fatty acids into fish as well? And one of my interesting findings was that while primary productivity did decrease mercury concentrations in the fish, it also did decrease the amount of EPA present I was looking at big head carp in those lakes. So it's, I think, another area of future research that we do need to look at is how these changes that we're seeing in productivity of our lakes also influence some of the beneficial aspects of our fish.
Okay. So I'm going to give one last chance for questions, and otherwise, so I'm going to count 5 silently to myself.
And I should have given my email address if anyone wants to reach out to me. I'd love to hear from you as well.
Okay, and I can provide that if anybody wants to, but Okay. Well, anyway, thank you so much, Roxanne, and thank you all for joining us. I really got a lot out of this, and I thought this was really interesting work. So thank you again, and we'll get together in a little while.
Thanks everyone for your time.
It was a great
Thank
talk.
you. That was very good. Thank
Thanks.
you.
Thank you.
Thank you very much.
Thank you.